Well,
You don't
other
want your
people
face to
might
be
not
seen.
want my face be. I could spare
So
the population that, but I don't mind.
you're fine. So welcome everyone. This is a talk I've been waiting for a long time because I've seen George talk before and he's invariably engaging, and it couldn't be more timely. So we're talking about plants as vehicles for vaccines, and of course we're still wading through the mire of the COVID pandemic, and you've got other things like monkeypox coming up. George has kind of been, I hope he doesn't mind me saying, a big hitter and a legend in this area of vaccines from plants. And most of his career, actually all of his career really, the hard end part has been at the John Innes Centre. Like me up here at JHI or SCRI, we've been here man and boy in our own institutes. But George may be a bit better than that. He's had a few breaks going to Cornell and Scripps Institute, not shabby organisations, it has to be said. And of course, he was named BBSRC Inventor of the Year for his work on plant-made pharmaceuticals. So we look forward to an engaging talk, and I've had a glimpse of some of it, and it is really entertaining. So it only leaves it for me to say over to you, George, and we're looking forward to it.
Well, thank you very much, Derek, and hopefully we'll actually get to meet again in person soon. um, well, hello, good afternoon, everyone. Hope you can hear me clearly. I'm going to talk, as Derek said, about plants as vehicles for vaccines. Now, I'm based, as again Derek said, at the John Innes Centre, which is in Norwich in the UK. And there's a photo of it in the bottom left. The fields around there are looking a bit greener than they do at the moment, I have to say. We really haven't had much rain around here for quite a long while. Anyway, so the John Innes Centre is a government-sponsored institution, a bit like the James Hutton Institute, as an equivalent, and it works on plant and microbial sciences. So first question is, and I'm sure many of you know the answer to that. I mean, it's a well-known term, but what is vaccination? And to get a definition, I turn to that font of all knowledge, Wikipedia, which says basically vaccination is the administration of antigenic material, that's immune stimulating material, to stimulate an individual's immune system to develop immunity to the pathogen. So you have a disease and you give the recipient, and it could be a human recipient or it could be an animal recipient, something which won't infect it but will stimulate the immune system and to make antibodies against the actual pathogen. So you're not having to infect to get this. Now, I mean, this has a long history, and in fact the whole name was coined through some work by someone you probably know very well, called Edward Jenner, who lived in the West Country in Berkeley in Gloucestershire, and he first did his experiments on vaccination in 1798. So it does have quite a history, and the term comes from the Latin for cow because he noticed that milkmaids in his area had developed these pustules on their hands associated with interaction with cows and milking them. But they also never seemed to get smallpox, which was a terrible disease prevalent at the time. And he rationalized that this somehow protected them. Of course, viruses hadn't even been described at that time, wasn't until another 100 years so they were identified, but it was just known as a disease. And he actually inoculated this, well, I mean, it's rather strange kind of statue. Oh, it's requesting to annotate the shared content. Oh, I don't know quite what that means. Shall I approve that?
We can't see anything, so we go ahead.
Okay, I'll approve it. Oh, I see what it means. I've got some tools. Right, that's an excellent idea. Pen tool and a pointer tool. Right. Oh, let's find the right one. Line, text tool, arrow pointer. Excellent. That's better. I've got an arrow here. I think you can point to. That was an excellent idea. Okay, so these pustules are here, and he then inoculated a young lad called James Phipps. History records, and this is a statue of him doing it. It looks a bit like child abuse to me, but some kind of initial vaccination and protected the local population against smallpox. Rather remarkably, I've even seen what claims to be the original hide of the cow, which called Blossom, which gave oh god, I've got some odd things coming up here. Gave the Sarah Neems, the milkmaid, the actual cowpox, and the hide is in St George's Hospital in Tooting. So go down here. Oh, I seem to have got something odd going on here. Let me try and remove this annotation device. Got too clever here. Let me just try and let me cancel or allow. No, I shouldn't have done that, I don't think. I'm struggling a bit now. You
You're
can
okay, just carry on, press the next slide, you should be fine.
okay. I could actually do that. I seem to have lost my ability to control my own screen at this point, which is
Try
frustrating,
using
to
your
say the
arrow
least.
key.
Oh, well, I'm trying to think where I can do this. I got too brave, I think. I've somehow lost my arrow, my cursor.
Oh, try using your arrow key on your keyboard.
No, unfortunately, that's not. Everything seems to have been detached by my doing that. Oh, may have to just see. Actually, I can't do a thing.
You could try stopping sharing, then resharing.
Yes, it seems to have just absolutely. I mean, I've lost all sorts of control here. I no longer control the screen. That was unfortunate. Okay, let's try again. I think like I should be able to be okay here. Yes.
Sorry?
Sorry about that. I got too brave with the technology. Okay, so you're trying to administer some immune stimulating material, and but the question is how you make it. Now, it was a live vaccine that Jenna really used, and that was really the basis of how you made vaccines in the initial years. And basically, what you'd have to do is identify the disease-causing agent, and this could be a virus, or it could be a bacterium, or even a fungus. Grow it in culture, get it purified, purify the particles or the material, weaken it, otherwise known as attenuation, and effectively cowpox is an attenuated version of smallpox, or inactivate it, kill it, and then administer the material. Now, the problems with this kind of approach, which has been very successful over the years, and I mean, classic vaccines that I was administered as a child, such as the Salk polio vaccine, is a killed vaccine. More recently, the Sabin oral vaccine is an attenuated version. This approach has been very successful, but it does depend on you having to be able to culture the pathogen, which can be difficult to get the conditions right. You also need to be able to grow a large amount of it, and it's potentially dangerous. Material safely, so a lot of containment. Inactivation, if you're going to kill it, has to be just right. And what I mean by that is, if you really inactivate it, really kill the pathogen, say it's a virus like polio, you'll actually destroy all its immune properties and it won't be any use. But if you don't do your inactivation quite thoroughly enough, there'll be residual infectivity. And so there's a danger of actually giving the disease. And live attenuated virus, such as the Sabin oral vaccine for polio, can revert to virulence because they're still alive, and that does happen. And of course, the whole thing can take quite a long time if you're identifying a new disease. And of course, we're thinking a lot of SARS-CoV-2 at the moment. So, I mean, this is used, and every year we go through the influenza vaccines, and for seasonal flu, and it's a really complicated process. You basically have to identify the strain, basically make an attenuated egg-adapted version of it by reassortment with influenza, and then grow it in embryonated eggs. Then, of course, it's a live virus, a live influenza virus at that moment. So you then do some what they call splitting or inactivation to dissociate the particles and then pack it. You get a timeline from January to October, and so it takes a good 9-10 months each year. There's a race between when you identify an emerging strain and getting a vaccine ready, and often you're a bit too late, and you haven't quite got enough to supply it, the population who needs it. So it's even with a well-developed system like influenza, which you know the properties of the virus, you're going to have to do this every year. It still is quite a long process. So the more modern method, and these methods have been deployed very much during the current SARS-CoV-2 pandemic, is to identify just those portions of a pathogen, a virus or a bacterium, which can give an immune response. So rather than have to grow the whole thing, then use recombinant DNA techniques to express these proteins, in this case in culture, and then purify the material. Or the modern alternative, which I'm sure everyone's heard about with Pfizer-BioNTech and Moderna, you can make a synthetic RNA corresponding to that protein and use the body itself to make the protein. So you actually inject RNA into the recipient and make the recipient make the protein rather than make it in some kind of other culture system. And then you administer the material. So that's the more modern approach, and it short circuits the time largely because you don't have to grow or handle dangerous material. So whole systems of ways of producing this material have been developed over the years. So why would you even think of using plants? Well, they produce large biomass. You can get a lot of material. Obviously, farming is largely based on this. They require minimal input. So, their energy source is the sun, carbon dioxide from the air for carbon, nitrogen from the air for your legume. So they don't require an expensive diet. The interior of the plants itself are naturally sterile, so you don't have to worry about treating it with steam or disinfectant. You won't have a problem with cross-contamination with animal pathogens because they don't grow in plants. And that's a problem if you're using things like mammalian cells. They can get contaminated with adventitious pathogens. Also, there's raised the possibility of edible vaccines. I mean, obviously we consume plants and we eat them. So maybe you could actually use an edible plant. That's been very attractive over the years. And so basically the hope, and all this work started probably around 30 years ago now, was to reduce the cost of protein-based pharmaceuticals by producing them in plants. Now, in case plants seem so far out in left field, just a very brief reminder that, of course, they are currently used to make pharmaceuticals, which are widely deployed. And examples are morphine and its derivatives are still mainly extracted from poppies by the ton. And they're, of course, very important for sort of opiates and treatments for pain relief. Vinblastin and vincristin, which are anti-cancer drugs, are extracted from the leaves of Madagascar periwinkle plants. Artemisinin, which is an anti-malarial, is extracted from Artemisia annua, which is sweet wormwood. And so plants have historically been used and are continuing to be used for small molecules, metabolites. But actually, the pharmaceutical industry is looking for ways of producing these alternatively, like understanding the pathway of synthesis of these and then introducing that pathway into, say, yeast, because you get a bit more control over the whole process. And there are various degrees of success on that. So could plants be used to make protein-based pharmaceuticals? Things that we normally associate with vaccines. I mean, those small molecules are more for treatments. So vaccines you could make, and particular virus-like particles, which are mimics of a natural virus. You could use them for that, and that's what I'll be talking about today. But you can also use this to make antibodies for immunotherapy, cancer treatment, or passive immunization, and with this treatment for COVID infections using antibodies to mop up virus within the body. You can use enzymes, make enzymes for enzyme replacement therapies, things like Gaucher's disease, where it's a lack of an enzyme in the patient, the sufferer, and they can be kept in relatively good health by supplying enzymes exogenously. So you have to keep on supplying them. But the question is, what advantage would plants have compared with other expression systems, such as bacteria, E. coli mainly, mammalian cells, yeast, or insect cells? So the idea basically is if you want to use plants, you could go from what is a rather complex type fermenter, although this is a very simple lab scale one, to what would be a rather simpler version of growing plants in a field. Now, this is rather fanciful that you'd ever grow the plants actually in a field, but this was a view about 1990 that this might be possible. The reason you would never really want to grow them in the field, even though you'd get massive scale-ups, one assumes, is that you'd have very little control of, other things landing on your plants. Other contaminants. So you'd always view them nowadays as growing plants under more contained conditions. So a lot of vaccines, many recombinant vaccines, are based on what are known as virus-like particles. These are very good mimics of the natural viral structure if you're going to make a vaccine against a virus, which is what I'll be talking about. The great thing about virus-like particles, as it clues in the name, they have a structure similar to the original virus, but they're non-infectious. Their size is just about right for them to be recognized by the immune system and taken up. And also they have this very repetitive structure. Again, because they so closely resemble the native virus, the immune system recognizes as an unwanted invader, and they stimulate aspects of the immune system. They can also be used in nanotechnology. And there's this well-known review now getting a bit old, which just shows that VLPs up here as a vaccine, they're really very good for efficient entry into lymph cells and uptake by antigen presenting cells. So they're just very good. They just look so like a pathogen, because they are very like a pathogen. So how do we make these things? Well, we use a technique called transient expression. And now many people may be very aware that you can transform plants stably to make different varieties. So maybe in time to time there is in the news, such as Roundup Ready soybean, where you make the soybean resistant to the herbicide Roundup, so you can spray the crops with Roundup and all the weeds will die, but not the soybean. BT cotton is another example where you make a permanent line of plants which have an extra gene or genes as part of their natural component. So it's a heritable system. This is not so much. We're using all the same systems. But what we're going to do is introduce the sequence into ordinary wild type plants. The plant we're going to use mostly is a relative tobacco, sometimes called Australian dwarf tobacco, because that's where it was first discovered, and its full name, Nicotiana benthamiana. However, the introduced sequence does not modify the plant germline, doesn't modify the genome in a heritable manner, but expression occurs very quickly in a matter of days after introduction, and therefore it can be very responsive to changing needs, this method. And it's essentially a batch process. So this is how it works, and I think I've got this. I'll go through it as I go, the basics of what transient expression involves. Oops. Place. So basically, it starts off with basically cloning 101, where you take the genetic structure of a pathogen, you take the bit of it which encodes a system capable of eliciting immune response, you insert it into a plasmid, and you grow up that plasmid initially in E. coli, so very straightforward recombinant DNA techniques. And once you've got what you wanted, you then transfer the desired plasmid to a bacterium called Agrobacterium tumefaciens, shown here growing on a plate, so it gives colonies. And this bacterium normally lives in the soil, causes a thing called crown galls. It's a motile bacterium, has sort of flagellae. and is chemotactic, therefore it will move towards plant tissue naturally. And you take the bacteria and your plasmid and give them an electric shock, and the plasmid goes within the cell, and it actually is amplified. And the bacteria are fine, they continue to live. And you can grow them in culture and then simply take the suspension up in a syringe. And it's as easy as this. You can just push the solution under the epidermis of a leaf, and it looks a bit soaked when you do that. See the area? And then once inside the plant, the bacterium gets to work. So here inside a leaf, see the cells, and the bacteria swim towards the cell surface because they recognize it, put out a tight secretion system, and transfer the plasmids into the cell. And they get coated with various proteins, which transfer them to the nucleus, this rather sun-like looking object. The plasmids get taken in there. Once in there, the DNA begins to get integrated, gets transcribed into RNA, starts making the protein subunits here, and the sun goes supernova, and all the RNA and subunits appear in the cytoplasm. And this, as these are shown as a viral subunits, they tend to be able to self-assemble. And was there purple? That part of the plant goes purple, and you get your particles out. And this will all take place the bit in the plant takes only about 6 or 7 days. You can go through this whole cycle of making synthetic DNA, cloning it into the bacterium, and you can get your particles at the end. It's all in about 4 weeks from start to finish. The process is scalable. You can do a syringe type infiltration here, but you can turn the process around and suck the bacteria into whole plants, and that's an industrializable process. And you can do huge batches by this vacuum infiltration method. And so you can get various scales. And we use plasmids, of course, that's the method we use developed here. Oh, it's getting on for 14 years ago now, called CPMV-HT, which is hypertrans. We just took a plant virus, showed that we could make a synthetic RNA and express the green fluorescent protein. And if you do your inoculation on a small scale, you can get GFP expression in the inoculated bit, but you can also control the amount you get by altering the sequence of the untranslated regions of the RNA. So you can have a controllable way of doing this. And you get, you can vary the yield of your protein for about 15 to 20 fold. And you might say, well, why do you want a low amount? Well, some proteins are toxic. So you can't, if you put them at a huge amount straight away, you're probably going to kill the plant. And there's a whole vector series here where you can do your cloning quite easily, put multiple proteins on the same plasmid. And again, these are quite old now. I mean, 2009, these plasmids was published. And just to give a brief outline of how you can do this in 4 weeks. This is an experiment that a student did on a rotation in the lab. Took the coat protein from a fish virus called nervous necrosis virus, did all the cloning in E. coli, transferred to agrobacterium, did the agro infiltration, and got her particles within the time of her rotation in the lab. And actually, we went on to show that those particles will protect fish because this is an important disease in aquaculture. So it is a quick process. It can be responsive. So we've done a lot of made a lot of different virus-like particles. Quite simple if you're using non-enveloped viruses, and things like bovine papillomavirus, which is important veterinary disease. Particles of another plant virus used in nanotechnology, a little nanocapsule. Nervous necrosis virus, the one I've just talked about. Hepatitis B core particles, which are very useful for display of extra antigens, as well as being an important disease in itself, hepatitis B. We've used particles of cowpea mosaic virus, which are useful in nanotechnology. Polio virus we've used, and that's the one I'm going to talk about today, because it's been in the news because of the discovery of polio virus in in wastewater in the UK. Oh, and we've also worked on a veterinary one called Pluton virus. Okay, so production of poliovirus-like particles. I mentioned that, well, isn't polio eradicated? I think that's what surprised people, actually, hearing in the news that, well, it's gone, isn't it? Well, not quite. There's been a very successful WHO program at really suppressing polio. And so there are only a couple of hundred cases a year, down from thousands in the 1990s. But the problem is existing vaccines tend to mean that you can't get rid of the residual level of polio infection, particularly the live attenuated Sabine vaccine, because it is live, it will continue to spread in the environment, and it does tend to revert. And that's why you're always going to get some floating around. And the problem is, the worry is that if you stop vaccinating, all these revertants will come back and start giving people paralytic polio. So can we find a different way of vaccination to replace this highly effective live attenuated vaccine? Well, yes, you can make VLPs, and you think, well, that would have been done years ago. But in the absence of the poliovirus RNA, these virus-like particles are relatively unstable, and they go to an immunologically irrelevant form quite quickly. This thing, the C form, or sometimes native to H, it's called, same thing. And so what you want to do is, can you make stabilized particles, and can you make them in plants? So with help from immunologist friends and people at the National Institute of Biological Standards and Control, who could handle live polio, they identified several stable mutants of poliovirus and told us what the sequence of these variants was. So it's a real clone by phone example. And to make the VLPs, you just have to take the region encoding the coat proteins, and that's a precursor of 4 different coat proteins, and put them in one plasmid, and the protease needed for their processing to make particles, and then co-inoculate the two. Now, the critical thing is, if you inoculate with the same amount or express the same amount of protease as the precursor, the leaves do indeed fall off the plants because the protease is toxic. But because it's catalytic, it's enzymatic, you can reduce the amount and still get particles made. And if you do that, you can get nice polio particles here. And you can solve their structure. This was done at the University of Oxford. Show they're empty, so they have the authentic D-type or N-type structure, and then you can use these particles to protect mice from challenge with poliovirus. So that's the basis of a potential vaccine, and that work's being taken forward by the WHO. The initial work was done in plants. Whether the final production system will be in plants is probably less certain. Most likely it'll be in yeast, but plants played a prominent role in getting to that point, identifying what you needed to do. Okay, so these are also known as non-envelope viruses. They have a protein shell encompassing nucleic acid. But what about envelope viruses, which are more complicated? And the reason I bring these up is, of course, SARS-CoV-2, causative agent of COVID-19, is an envelope virus. And many other important viruses are enveloped. They're more complicated ones. They contain host components, particularly the membranes. They have envelope proteins, which have glycan sugar residues on them. Maturation often uses a host-encoded protease. So the host has to, it's an interaction between the host and the virus, and different cellular compartments are used for glycosylation and processing. Now, to try and mimic this is potentially a bit more complicated. Fortunately, it turned out with SARS-CoV-2 not to be too bad. So this is a rather old pattern, the way this looks. So the one second from the top is the old name for SARS-CoV-2, and what you need to do, there are 3 structural proteins, but actually the only one I'm going to really mention is the S protein. That's the spike protein, which gives the particles their very characteristic look of a crown, hence the name coronavirus, and they form trimers on the surface. There are other ones which are will be in the particles, but we haven't really worked significantly with those. But everything concentrates on the S protein that has most of the antigenic determinants. And so to make these implants, you'll need, well, we just use the S protein eventually. We have done work on the two other ones. Maybe the M protein, we haven't had to use that. It's also membranes that's from the host. Glycosylation apparatus, that will be host encoded. All the SM and SM as well as M and E are produced from separate subgenomic RNAs. So there's nothing fancy about processing, no protease processing needed for this. And there's evidence that some of the antigenic sites are actually involved in distal parts of the protein being brought together. So you want a structure. So expression of S protein alone is sufficient to give VLPs. And this just shows this is a blot which just shows the amount and we can measure the amount of material. This is just, actually, if you try and co-express the other two proteins, you reduce the yield because they're a bit cytotoxic. Here you go, you get a yield, you can measure the amount. Most importantly, a picture paints 1,000 words. Here's an extract from plants. You get these nice looking coronavirus structures which self-assemble within the cytoplasm. You can show that these particles are recognized by serum from recovered patients, COVID patients, but not by pre-pandemic serum. So these are antigenically relevant. And we've been doing a number of experiments showing these will bind to the SARS-CoV-2 receptor, a thing called ACE2. But the main reason for us making this, because we're not really a vaccine producer, is actually to get a better understanding of SARS-CoV-2 and coronaviruses in general. The problem is with working with these things is you need category 3, high containment facilities. Whereas working with a plant-produced material, as long as it's authentic, you can actually say it's much cheaper to work on, and you can do a lot of studies you can't easily do. So we are mainly interested in understanding the basic biology using these, but you can certainly make them. And so the question is, can you commercialize any of this plant-based stuff? And essentially the two different ways of doing this from our point of view: licensing agreements with companies, pharmaceutical companies who might be interested in the technology, and also setting up your own facilities. And I'd say we've done both. So we have a collaboration with Medicago. This is quite long established, initially with influenza virus. Now Medicago are now part of Mitsubishi Tanabe, and they've reported positive phase 3 efficacy studies on a quadrivalent influenza vaccine. So this is for seasonal flu. There's hopes that could be on the market. Of course, all talk about flu has been a bit sort of in the background since the COVID pandemic. But in December last year, two Medicagos, there's only one, and GlaxoSmithKline announced positive phase 3 efficacy and safety results for adjuvanted plant-based COVID-19 vaccine candidate. So that's a vaccine that looks promising. Next question though is, are these better than equivalent vaccines? Because of course lots of people, and I mentioned RNA vaccines earlier, have been making COVID vaccines. And so the plant-make anti-COVID vaccine called Covivence has been approved for use in Canada, and it's based on a modified version of the S protein made by transient expression in N. benthamiana, which is very similar to what I just showed you. Ours was done on a small scale, using a non-modified version. But then if anyone followed in the news, there was a little bit of a dust-up, because part of one of Medicago's shareholders is Philip Morris International, which is a tobacco company. And WHO have been reluctant, I don't know if it's been resolved yet, to put this plant-made material, approve it as on their list because of the involvement of a tobacco company. It's the kind of thing you might not really think about in the first instance. It's not the fact that the plant, it's a tobacco plant, it's the fact that it's part of the tobacco industry. And that's caused a few eyebrows, I guess. It might be resolved, one hopes so. So the alternative to that is, rather than licensing your technology, is to build your own facility. And this is something I suggested, going back a few years now, so around 2012, 2013. So you want to have a facility to grow your own plants, mechanism for efficient agro-infiltration of plants, a facility for extracting of your products, efficient way of treating and disposing of waste. And this led to the creation of this company and facility called Leaf Expression Systems here in Norwich, adjacent to the John Innes Centre. And this is just a little bit about that in a rather, I hope, rather humorous way, going from when I first proposed it actually at a meeting in 2013, and then what happened subsequently. So here we go.
Previously at an ASM.
I have just created something totally illogical.
That's what I like about it.
If you build it, he will come. If you build it, he will come.
If you build what, who will come?
You didn't say.
I hate it when that happens.
Me too. Did you hear it?
Go the distance.
Yes.
Unbelievable.
It's more than that. It's perfect.
Sometimes, when you believe the impossible, the incredible comes true.
And now, the reality.
And that facility, which is a genuine one, that's not a mock-up, that's actually a time lapse of the construction, was opened on 23rd January 2017, so more than 5 years ago, and actually opened by someone who probably looks vaguely familiar. That's actually Boris Johnson's younger brother, Joe Johnson, who at the time was Minister of Science. So the last question I'm going to leave you all with is plant-based production competitive with other approaches? So yes, there are a lot of approaches out there, including this, expression of proteins in other systems, RNA vaccines. Can it compete? But even my attitude is exemplified by poliovirus work. If plants are not used as the final production system, can they contribute to the development of new pharmaceuticals, new vaccines? Because they're so easy and quick to use. And so you can check whether something looks promising and then go into a more traditional production facility. And with that, I won't belabor all the acknowledgement. There have been a lot of people, both working in my own group, working with the Poliovirus Consortium, which I talked about, and of course funding from various agencies, including the research councils and the EU funding as well, World Health Organization. So thank you very much, everyone. I hope that was okay.
Thanks very much, George. And I imagine everyone will just show their appreciation as we normally do. I think that's clapping hands anyway. So is there any questions from anyone?
Should I stop sharing my screen? Would that be a good idea?
Yeah, that's cool.
Yeah, I can see people now. That's the problem.
That's the right one. Abby, you have a question?
Hi, George. Thanks very much for your talk. Fantastic. You mentioned that protease, when you overload the plant with the protease in the polio vaccine, you kill the plant. Have you tried using a system where you can actually induce the protease once the other parts are made?
What we found was actually easier to do. That's certainly one approach you can do. But we simply altered the sequence of the 5' UTR to downregulate the expression. and did co-infiltration. But having an inducible system, you can imagine, we have looked into that using inducible promotion. That's certainly possible as a way. There are other people who've used ribosomal frame shifting successfully. Obviously, so you get a lower amount of the product downstream. So that has been used. But using an inducible system is definitely possible.
Thank you.
Rob, you want a question?
Yeah, hi George. Thank you for the presentation. Now, anyone that's ever tried purifying a protein from plant material will have horror stories of the difficulty it can sometimes present. I mean, is this an issue in plant production of vaccines, and how do you overcome it?
Yeah, I mean, Rubisco is ubiquitous. So actually, we're fortunate. Oh, no, just my seat a bit. Sorry, I've been sitting in the same place too long. Fortunately, with the virus-like particles, they're of a size that isn't so much of a problem, because although of course Rubisco is a complex as well, you can actually fractionate quite readily on the basis of size, and you can do it either by ultracentrifugation or ultrafiltration. So with that, but if you're talking about soluble proteins, which we haven't worked with as much, yes, absolutely, that can be a total nightmare. One of the interesting things, actually, which I didn't go into, was you also get cross-linking of proteins within plants when you extract them. And this was a bit of a mystery for a time, why some of the plant-based VLPs seem to perform slightly better than their yeast or animal cell-produced equivalents. And of course, if you look at how vaccines are actually produced, one of the first stages people do once they've got the pure material is to partially cross-link it, to stabilize it. So I think the stuff coming out of plants is actually sort of pre-cross-linked to a certain degree, which may well explain that. But yeah, I mean, I absolutely take your point. It can be an absolute nightmare to get rid of all the other plant proteins, particularly Rubisco. But we're fortunate because of the size. We can basically use the size properties of the VLPs.
Okay, thank you.
Let me ask a question about comparing the transient system against the more stable GM approach. I mean, if you've got something like flu, the transient one's going to be more useful because it evolves. But going back to that one you were talking about the enzyme deficiency
Yes, yeah.
pathology, in that one, presumably, you could just keep a stable transgenic then, and that actually creates IP for the company. I'm thinking from a
Yes, you can certainly do that. I mean, the people who've done a lot of work actually on the sort of Gaucher's disease, this thing called Eleliso, this cerebrosidase, is a company called Protalix in Israel. Now they've used cell culture rather than whole plants. They started off with carrot cells in disposable sort of bags, and this, of course, it seems a bit sort of obvious, I suppose, but the wonderful sort of orange cells in hanging bags. But so they've used those, and they find those. It's the level of expression I think they like. Because one of the problems with a lot of stable ones, not universal, is that the actual levels of protein you get can be quite low. You have to process a lot of material, whereas you can get much higher levels transiently. But I can actually absolutely see for those, if you get high enough levels of synthesis, a stable system would almost be ideal. Now there is a company who works on moss, and they're transgenic moss, and they grow them in these lighted tubes, and they're making enzyme replacement therapy materials, and those are stably transformed. So I can certainly see stable transformation for those things as being a very viable option. What I should also say is that often transient expression and stable transformation sort of go hand in hand, because they use essentially the same constructs. And a lot of people, what they do is they test their constructs first transiently, and then when they think they've got something that they want, you then go through this much more laborious business of then integrating it into a stable line rather than having to do that first.
Okay, any other questions? If not, I've got another. Abby, did you want another one? Go ahead, you're on mute.
There you go, technology. Sorry.
Well, so what happened to me?
Exactly. In large-scale production, using transient expression, do you think there is a scope for improving transient expression? And what are the stages or the elements that still need optimization, if there are any?
Okay, now that's a very good question. There are a number of elements which people are addressing. So because it's a batch process, you want to get the maximum synthesis you can. And so people are still working on refining the plasmids, double promoters, better terminators, etc., to try and say you get the maximum amount in a given weight of leaf tissue. Also, another thing which, on the smallest, obviously the classics on the lab scale, you don't really think of, but it's critically important for industry, is the expense of growing all the agrobacterium. So we just put as much as we like in. It's got a syringe full, so what?
Yeah.
But it becomes a major issue. So people are looking, what's the lowest amount, lowest multiplicity of agrobacterium you can use? Plus, are there strains which are more capable of transferring efficiently? And so there's that side of things, the infiltration side being looked at as well. And of course, always better ways of purifying. I mean, I had a question about the proteins contaminating, because one of the things early on is that people, you don't get from, say, yeast, sort of not the same way, but for yeast or bacteria or certainly animal cells, is all this incredible amount of cellulose which tangles up and blocks filters and everything. So there's been a huge amount of work on the downstream processing side, to avoid all this clogging up of material.
Thank you.
Okay, maybe I can just ask the last one. So you've been in this game for a wee while, George. So how do you think it's up on the increase? Because my gut feeling is, certainly from the commercial perspective, it is.
Yes, I think it is, and a pun here, but it's one of those. It's an ill wind that the latest, the COVID outbreak had caused a bit of a shot in the arm for people working on vaccines, which had been sort of slightly pushed into the background. I mean, everyone got vaccinated, but there wasn't a huge amount of new stuff coming out because everyone said, "Oh, we've got everything we need." Well, obviously that wasn't true, and people are now far more aware that something new can come along, either something which was known about but had a very localized distribution, has suddenly moved out of that, or to something, a variant, a zoonotic disease such as SARS-CoV-2. Also, we have this monkeypox and Zika before that. So the people are becoming more aware that you need to be able to respond quickly to these things. So I think methods for vaccine production are on the up, and people are sort of very much interested in it. And plants, I think, can play a role in it.
Is there much of a feel for people using it or thinking about it for inoculating animals? So because I know there's huge problems with it, like in China and livestock, the pig swine fever. I don't know if that's a viral one. I'm not sure.
Oh yeah, no. There's a very a lot of interest in that. In fact, the work we were we were very fortunately well placed to work on SARS-CoV-2 because I had a visitor from Korea who came here to work on porcine epidemic diarrhea virus, very important disease in the Far East, and it's a coronavirus. So he'd already done some initial work showing what you needed to make a VLP, and then SARS-CoV-2 hit. So we know how to do this in theory. Obviously, you have to change for various aspects. So no, I think there is a very large thing, and its real hope is, can you get an edible vaccine?
Yeah. Okay, that's a nice point to end on. Well, thanks very much for that, George. And I feel a bit ashamed that I didn't realise corona meant crown. And actually, when you see the picture, it does make it a bit bloody obvious. Hopefully everyone's learned that one, but hopefully everyone's enjoyed it. It's been very engaging, and hopefully we can meet again.
Okay, in person next time. Cheers then, everyone.
Thanks, everyone. Bye.
Bye-bye.